Good morning, everyone. I'm very happy to present you our company, SK Pharmteco. In fact, we are part of the second in size South Korean conglomerate, SK. So SK, this is a big conglomerate active in biotechnology, chips, batteries, and semiconductors. That's 215 billion in assets, and that's a revenue of 120 billion of dollars. And in fact, within SK, you have SK Pharmteco, which is a CDMO active initially in the production of active pharmaceutical ingredients, but which has recently invested into the cell and gene therapy space. Huge expertise in APIs. This is more than 70 years of expertise. Actually, the company is divided in 3 different business units. Small molecules with sites in the US, South Korea, and Ireland. The cell and gene space division with 2 sites, one in the US, one in France, Europe, which is today, it's more than 500 employees and 115,000 square feet of global footprint. Last, analytical, we have a specific division for analytical testing, which include both APIs and testing for cell and gene therapy products. This is our global presence, so 3 sites in the US, California, Texas, and Virginia for small molecule, Dublin in Ireland, and 2 sites in South Korea for small molecule. For cell and gene, we have a site in King of Prussia, west of Philly, US, and a site in France. A few words description of our current capacity. So what you have here is the site in France, which in fact, what you have on the top right of the slide is our original plant, which has been commissioned roughly 10 years ago, so 2013. We are very active and expert on the site in the development of processes for viral vectors with 2 specific platforms, which I will describe later. One for the lentiviral vectors and the second one for the AAVs. We have recently, in fact last year, completed the construction of a second manufacturing site, which is really designed for large-scale manufacturing, commercial manufacturing of batches, and a site that we have designed to be very compliant with both regulations, EMEA and FDA. So this site is actually under qualification, and so will be ready, commercial ready, by the end of this year. Now, we have a site in the US, King of Prussia, so close to Philly, which is a very impressive site, very large, where we do have today capacities for viral vectors, cell therapy manufacturing, plasmid production, and a very impressive range of analytical testing capacities. So this site today is fully set up with GMP manufacturing capacities, process development with a huge capacity for process development and also for process characterization of GMP processes, and a real state-of-the-art capacity for testing. So we have the latest technologies for product characterization of viral vectors as well as a cell therapy product. What I would like also to stress is the digitalization. As you perfectly know, having electronic batch record, a full electronic traceability of the processes, this site is today fully set up with a very nice digital footprint actually implemented. So, in other words, today, SK Pharmteco, we have the possibility, we have a quite unique offer, a comprehensive offer of solutions really to support our client projects from A to Z, with nucleic acid, so plasmids, viral vectors with multiple platforms, LV, AAV, but we do also have expertise with oncolytic viruses and adenoviruses also. Cell therapy manufacturing, huge capabilities in analytics, and last, and that's also very important in our business, we provide regulatory support to our customers in terms of CMC. So with a team of experts which really help you really to design your development and help you to make sure that all the development we're doing are in line with your clinical objectives. A few words about two platforms which we have branded. On the left-hand side, Lentisure, which is really designed for the production of lentiviral vectors. And on the right-hand side, AAV Velocity, which is designed for the production of AAVs. And in fact, what does a platform mean for us? This is a very identified process. We have, for both platforms, proprietary cell lines, which we have developed internally. We have high-producing cell line. It's also a proprietary transfecting agent, and the combination of the cell lines, the agent, and these defined steps really give us, and I will show you examples, very high process yields. With those platforms, our commitment is to deliver a released product within 12 months, from the project start up to the release, ready to use in your clinical trial of our drug product. Few examples of the productivity of those platforms. What you have on the left-hand side is results from our Lentisure platform. What you have here, we have the productivity of this platform across different scales, from 250 milliliters, 10 liters, 50 liters, and up to 200 liters suspension process. So we have demonstrated really the scalability of the process across the different scales. We have also demonstrated that those yields are obtained with different transgenes, is what you have in blue on the bottom left. The two last scales, the 50 and the 200 liters, are today developed under GMP, so you can have GMP products coming from those scales. On the right-hand side is examples of the AAVs platform. On the top right is results with an AAV8 with a specific transgene, and on the bottom right with an AAV9. So with this platform, what we can achieve is titers up to 2 times 10 to the 11th viral genome per mil, which we have demonstrated across different scales, very small scale up to 400 liters, and we have very, very recently generated data with a 1,000 liter bioreactor. Track record, very important with both platforms. We have a very substantial track record. So talking about the lentiviral platform, so we have produced over 100 batches, of which 40 for clinical trials with 14 different transgenes. For the AAVs, it's also more than 100 batches produced, 55 cGMP batches, with 5 different transgenes. And those batches have been tested both in the US and Europe. Now, beyond those 2 platforms, suspension-based, we're actively innovating and working for the development of packaging cell lines. So what I'm going to show you in these 2 slides is the development we're doing today with a US-based company, Asimov. So where really the goal here is to develop a packaging cell line for the production of lentiviral vectors. So we have included with Asimov the 3 helper genes into the cell genome. And what you have here is example of productivity under and with using different process conditions. And in fact, we have reached 10 to the 8 transduced units per mil with a given condition, which is very comparable with what we obtain with the transfection-based platforms. What you have on the right-hand side is very interesting, is that there is also the possibility to optimize the sequence of the transgene. So for confidentiality reason, I cannot disclose what's behind the bar, the graph you have on the right. But that's today a commercial CAR-T where you have on the red the productivity of the original sequence, and on the right the productivity after the optimization of the sequence of the transgene. In terms of stability, we have demonstrated the stability of this packaging cell line over 27 passages, which in fact makes us very comfortable that this cell line is today able to support large-scale process and definitely up to 1,000 liter scale. On the left-hand side, the 3 graphs show you the expression of the helper genes, and that's the titration of mRNA. On the right-hand side is the expression of the transgene. And as you can see, we have a stable expression up to 27 passages. I just would like here to complete. So, in fact, as a conclusion, SK Pharmteco is today really fully set up, really to provide a very comprehensive range of services. We are able to support the client from A to Z with multiple services from nucleic acid production, viral vector cell therapy, and an in-depth and extensive analytical offer. I would like to thank you very much for your attention. Very happy to take any questions you have, either right now or offline. And I wish you a great conference. Thank you very much.